Bismillah ar-Rahman ar-Rahim. This is the description of the cytology data show. This is the start of the data show presentation. We will start by the cell membrane. Here the cell membrane is formed of phospholipid bilayer, two layers of phospholipid. Each phospholipid molecule is formed of a head which is polar, hydrophilic, charged, and the tails, which are called fatty acid tails, which are non-polar, not charged, and hydrophobic. Here we have the protein. We have the integral protein, which is traversing the whole thickness of the bilipid layer, and it is called transmembrane protein. The integral protein or transmembrane protein help in the transport as they form the channel protein or carrier protein. Also, we have here in the picture the carbohydrate element, which is present only on the outer surface, either attached to the protein to form a glycoprotein or attached to the lipid, which is not present here in this picture, to form glycolipid. So this is the hydrophilic head, the hydrophobic tails, fatty acid tail, and this is the integral protein or transmembrane protein, and this is one type of the lipid component in the cell coat, which is the glycoprotein. Another diagram for the molecular structure of the cell membrane: the phospholipid bilayer, the head of the phospholipid and the tail, and here we will see the protein content. We have here the integral protein, which is passing through the whole or part of the lipid layer, but it is embedded in the lipid layer. This is called the integral or transmembrane protein. And also we have another protein, which is peripheral protein, which is outside the bilipid layer. It may be on the inner side or the outer side. As you see, it is a non-continuous layer, and it is present outside the lipid bilayer, and it is called peripheral protein here, as you see. And it can be easily removed from the cell membrane, not like this integral, which is not easily removed from the cell membrane. In addition, you will see this carbohydrate element formed of oligosaccharides attached to the protein to form glycoprotein, or attached to the phospholipid head to form the glycolipid. And as you see, the carbohydrates are only present on the external surface or the outer surface of the cell membrane towards the external environment, as they act as receptors. Here the EM picture showing the cell membrane of two adjacent cells. This is a cell membrane of one cell. This is the intercellular substance, and this is the cell membrane of the adjacent cell. Each cell membrane is formed of two dark lines or two electron-dense lines representing the hydrophilic portion of the phospholipid, and the central electron-lucent layer representing the fatty acid hydrophobic tails. And on the surface of the cell membrane, we have the coat, and this is the intercellular space, and this is the cytoplasm. This is the side of the cytoplasm. This is the cell membrane, two dark lines and a light or electron-lucent line in between, forming what is called trilamellar appearance or unit membrane. Another structure here will be the mitochondria, which is one of the membranous organelles responsible for the production of the energy in the form of ATP, and some of the energy is released as heat. It is formed of double membranes. The outer one is smooth, the inner one is thrown into shelves or crystal. Here is the EM picture, the two membranes, the outer one and the inner one, and of course the outer one is more permeable than the inner one. It has pores called porin, and we have intermembranous space in between the two membranes, and then we have the cristae arising from the inner membrane, and we have on the surface of the cristae we have the elementary particles containing ATP phosphate ATP synthase enzyme, which will form oxidative phosphorylation and produce the ATP molecules in which the energy is stored. And here we have the matrix. The matrix here contains the enzymes of Krebs cycle, which are oxidative enzymes. So in the matrix we have oxidative enzymes. In the elementary particles we have oxidative phosphorylation enzymes, which is the ATP synthase enzyme responsible for formation of the ATP. Nothing here evident except this matrix, of course, and here we have some electron dense particles inside the matrix. Here we have also EM of the mitochondria, the double membrane, the intermembranous space, the cristae, and the matrix. Again, we have here the double membrane, the cristae, the matrix, and we have here very evident dense bodies, which are nothing but calcium that act as a catalyst for the oxidative enzymes present in the matrix. In the matrix also we know we have other contents. We have DNA, we have the 3 types of the RNA as it can form its own protein, in addition to these dense particles and the enzymes of the Krebs cycle, which are oxidative enzymes. Here we have another membranous organelle, which is a Golgi apparatus. The Golgi apparatus is formed of flattened saccules arranged above each other from 3 to 10, and they are called saccules. We have an expanded pulse at the periphery of the saccules, and the saccules have a concave surface, convex surface, or this is the mature surface and this is the immature surface. This is the trans surface from which the secretory vesicles arise, and this is the cis surface which receives the transfer vesicles from the rough endoplasmic reticulum. Here we have the flattened saccules, and this is the cis which receives or in which there is entry of the contents of transfer vesicles coming from the rough endoplasmic reticulum. And these are the flattened saccules, and this is the exit or trans surface from which we will have the secretory vesicles arising from this mature surface. So this is the cis and this is the trans. This is the transfer vesicle and this is the secretory vesicle. Again, we have the flattened saccules from here we have more than 10 actually in this picture, but they are flattened, having expanded ends, as you see. And in the center of these vacuoles, we have a slight moderate electron dense material, and this is the surface from which we have the secretory vesicles. This is the trans surface or exit surface, and this is the cis which receives the transfer vesicle, and it is the entry surface. So this is the entry and this is the exit. This is the cis and this is the trans. Here we have Golgi apparatus in the light microscope, but here it is stained by its special stain, which is the silver. Here all of these are nerve cells. And we have here this network of granules here scattered in the cytoplasm, and actually they are surrounding the nucleus. So this is the Golgi apparatus in the nerve cell stained by silver. The nerve cells are more or less here rounded, and they are showing this reticulum of brown dots or granules, which are representing the Golgi apparatus, which appears as a reticulum surrounding the nucleus. The nucleus, of course, is not stained in this section, and you are only seeing these brown dots representing the stained Golgi apparatus in the nervous cells. Here we have the Golgi apparatus appearing in between the nucleus and the secretory side of the cell. All of this, it is the wall of the epididymis. It is lined by these tall cells, and these cells have the nuclei near to the base. And in between the nucleus and the top of the cell, which releases important secretions that will feed the sperms here in the lumen of the epididymis to be mature. Here between the nucleus and the secretory pole, which is the apex of the cell, we have this large brown stained material, which is the Golgi apparatus. So in the epididymis, which is an organ in the male genital system, important for the storage of the sperms and making these sperms mature by addition of secretions. These secretions are released from the top of the cells, so the Golgi apparatus is present in between the nucleus and the top of the cells, as the secretory vesicles will arise from this surface and will pass through the apex of the cell to reach the lumen. So this is a Golgi apparatus in the epididymis stained by silver. You should mention these three important informations: the organelle itself and its site, and the special stain, which is silver. Here, Golgi apparatus in the cells stained by hematoxin and eosin, it cannot be stained. But here, this cell has a basophilic cytoplasm or a dark basophilic cytoplasm, so the Golgi apparatus, as it is not stained, will appear as an unstained area in this dark blue background. So this is the site of the Golgi apparatus, which is not stained at all, and it appears by contrast here in this background, which is dark blue. So this is a negative Golgi image here in this cell. This cell, which is characterized by having the darkly basophilic cytoplasm, it is the plasma cell, which secretes the antibodies. These antibodies are glycoprotein in nature, so it has a well-developed rough endoplasmic reticulum, making the cytoplasm darkly basophilic, and a well-developed secretory, which is the Golgi apparatus. But here the Golgi is not stained, so it will appear white area in this dark blue background. Now we will move to another membranous organelle, which is the endoplasmic reticulum. The endoplasmic reticulum is of two types. One which is near to the nucleus, and it is studded with the ribosomes, so it is rough and it is called rough endoplasmic reticulum. It is characterized by having flattened cisternae, and they are covered with the ribosomes on the surface. So this is the lumen of the cisterna, and these are the surface covered from here and there by these electron-dense particles, which are the ribosomes, 20 to 3 nanometer in diameter. When we see these parallel flattened cisternae, even if you cannot see these ribosomes, it is the rough endoplasmic reticulum. Of course, you know that the endoplasmic reticulum, the rough endoplasmic reticulum, is responsible for protein synthesis formed by these ribosomes, its segregation inside the cisternae in order not to make a hydrolysis of the cytoplasm, and its initial glycosylation by addition of monosaccharide to this protein if the secretion is glycoprotein, and then it will pack this secretion and deliver it as transfer vesicle for the Golgi apparatus. Another picture. Here you can see that the cisternae are flattened, very parallel, and if you look carefully, you will see these dots on the surface. So this is the rough endoplasmic reticulum. And you should remember what are the functions of the rough endoplasmic reticulum: synthesis of the protein by ribosomes, segregation of this protein, and the package of this protein in the form of transfer vesicle to the Golgi apparatus, initial glycosylation, and it is an intracellular pathway in the cell. When you see the the cisternae in the form of these tubules, which are looking like rounded sacs, and they are not parallel at all and not flattened, this is the smooth endoplasmic reticulum, and they are not covered by ribosomes. So when you look for the shape of the cisternae, you can diagnose the type of the endoplasmic reticulum. If parallel flattened, it is the rough, of course, with the ribosomes. And if they are not parallel and not flattened, appearing as the form of sections in tubules, and of course devoid of ribosomes, this is the smooth endoplasmic reticulum. You should remember the functions of the smooth endoplasmic reticulum, which is the synthesis of the phospholipids and the steroid hormones, and formation of phospholipids of the cell membrane, detoxification of drugs and hormones in the liver, and also intracellular pathway. Here we have a picture of this organelle with the cristae. This is a mitochondrion. And here we have these tubules, which are not parallel and devoid of ribosomes. These are the smooth endoplasmic reticulum. Again, this is a good picture to compare between the rough endoplasmic reticulum and the smooth. They are here flattened and parallel with the ribosomes. This is rough. And here they are not flattened. They are looking like tubules or sacs, and they are not parallel, and they are not provided with ribosomes. So this is on the right side. It is the smooth endoplasmic reticulum. Here another picture, very nice, showing that we have, this is the rough endoplasmic reticulum and this is the smooth. Of course, the rough is towards the nuclear envelope and the smooth is towards the cell membrane, and they are intercommunicating. Very important to know that they are intercommunicating, but this one has its own functions and this one has its own functions. Now we will move to the primary lysosomes. The primary lysosomes, the lysosomes is a membranous organelle formed of one or a single membrane containing hydrolytic enzymes. These hydrolytic enzymes, you should remember what are these enzymes. We have here phospholipases, nucleases, proteases, sulfatase, and acid phosphatase. These lysosomes, they are formed, or they are the enzymes here are formed by the ribosomes on the rough, and then they are transferred in transfer vesicle to the Golgi, and from the trans surface or the exit surface of the Golgi, they are liberated, and they are not now forming any function. They are recently liberated. They are called primary lysosomes. The primary lysosomes are characterized by having homogeneous appearance, moderate electron dense appearance, and it is homogeneous in the electron microscope because it does not start now its function. After it starts its function, it is converted into secondary lysosomes of different types. Here's the picture for the different types of secondary lysosomes. Of course, you can here diagnose these are the mitochondria because double membrane and the cristae. This is also a mitochondrion. And then you will diagnose the following. This is the primary lysosome of homogeneous, moderate electron dense appearance. And here we have the appearance of a secondary lysosome in which you can see moderate and dark and light, so it is heterogeneous, different electron densities. Why? Because it is a secondary lysosomes in which we have digested and undigested material. And what is the type of this secondary lysosome? This secondary lysosome, it is called heterolysosome, which is formed by the fusion of the primary lysosome with a vagosome, which is the introduced solid particle inside the cell. After phagocytosis of the solid particle, we have a vesicle called phagosome inside. Fusion of the phagosome with the primary lysosome will form a secondary lysosome where we have a digestion here and heterogeneous appearance, and this second lysosome, it is called heterolysosome. Here we have also heterolysosomes. All of these are heterolysosomes because we have different electron densities. But this one is also a secondary lysosome, but in which we can see some droplets. So this one is a secondary lysosome formed by the fusion of the primary lysosome with the pinocytic vesicle. So it is a multivesicular body, or a type, it is one of the types of the secondary lysosome formed by the fusion of the primary lysosome with the pinocytic vesicle to form the multivesicular body. So we have here primary lysosome, heterolysosome, multivesicular body, secondary lysosome, heterolysosomes, and these are of course mitochondria. Here we have the multivesicular body also formed of, it is of course membranous because it is a fusion of the primary with the pinocytic vesicle, and it contains some vesicles here, so many vesicles here, so it is called multi, means many, vesicular body containing many vesicles inside, which is a type of secondary lysosome. Here also we have mitochondria, and we have heterolysosome or a secondary lysosome as you see. Here we have an interesting picture for a third type of secondary lysosome, which is the autolysosome. When the organelles like the mitochondria or rough endoplasmic reticulum became old and non-functioning, they are surrounded by membrane to form autophagic vacuole. Why auto? Because it is coming from the inside of the cell. It is my structure, not a hetero, not a different structure. No, it is my structure, so it is called autophagic vesicle. Autophagic meaning the phagocytosis of something from inside the cell, and this process is called autophagy. Here, the autophagic vesicle will unite with the primary lysosome to form a secondary lysosome like this one, containing this engulfed mitochondria. So it is called auto, means my lysosome. Autolysosome meaning a lysosome digesting something from the inside of the cell. As you see, it is an old mitochondrion.